Hello students, welcome to EPG Pathshala. I, Dr. Vikas Rana from Punjabi University, Patiala. Today we are going to talk on module entitled Characterization of Microemulsion from your paper Novel Drug Delivery System Part 2. Microemulsions are generally recognized as clear, transparent, thermodynamically stable dispersion of oil and water. They are stabilized systems consisting of interfacial film formed by surfactant and or co-surfactant system. The main advantage of this is they are having oil droplet size of 10 to 200 nanometer. Therefore, microemulsion delivery system need to be characterized so that we came to know that yes, we have prepared a very good microemulsion system. For these properties, we have to characterize some additional properties that made delivery system effective and useful. There is a need to develop such type of characterization techniques. So students, let's see what we are going to learn from this particular module. The main objective of this particular module is to highlight various characterization techniques used to evaluate and confirm microemulsion based delivery system. In another objective of this module is to learn various research activities conducted on the basis of these characterization techniques or related with these characterization techniques. So to begin with these characterization techniques, it is very important to know that how these samples of pre-concentrated microemulsion or you can say a reconstituted microemulsions are treated together. The pre-concentrated microemulsion is a sample which does not contain water. Yes, which does not contain water. It is only a concentrated microemulsion consisting of oil phase, surfactant and a co-surfactant mixture. Whereas if we transfer this particular pre-concentrated microemulsion and dilute it with water, and in different proportions of water, that may be we can take it as 1 ml pre-concentrated microemulsion is diluted with 10 ml, 20 ml, 30 ml, 100 ml of water. That depend upon the test for which you are going for. So that type of reconstituted microemulsion we will obtain. So therefore, what we can classify between these two category of micro samples for the characterization of microemulsions are the sample which contains water is called reconstituted microemulsion or diluted microemulsion, whereas the other one which is called a concentrated microemulsion or it is called as pre-concentrated microemulsion. The test on reconstituted microemulsion are first we have to examine the phase behavior of a microemulsion. That is, the phase behavior of a microemulsion is tested using visual inspection. That is, we can just see what type of the visibility of a solution is, that is clear or transparent or translucent. And this type of behavior can be quantitated by using transmittance studies employing UV visible spectrophotometric method. Another method is microscopy method, which is phase contrast microscopy. And last method is freeze fracture TEM. The physical characterization of a microemulsion is determined using particle size analysis method, thermodynamic stability method. Thermodynamic stability is very important and it can be determined by three methods. One is cooling method, heating and cooling method, that is. Then is centrifugation method, and one is cloudiness or cloud point determination method. Then is rheology determination and surface tension of a liquid pre-concentrated microemulsion. The morphological characterization of microemulsion are to be conducted using TEM analysis or SEM analysis, and electrical properties are determined by zeta potential, conductivity determination, and mobility of a microemulsion. The drug delivery performance studies of a microemulsion delivery system is determined by evaluating drug content, that is how much amount of drug is present or untrapped inside the microemulsion. Then in vitro drug release studies are to be conducted as per the standard pharmacopoeial methods or as per the guidelines which are given in the ICH, or ex vivo studies across biological membranes are to be conducted. These biological membranes may be permeation across Caco2 cell lines, or averted rat gut sac method, or permeation across PAMMA, that is PAMA membranes. In vivo pharma performance or in vivo pharmacokinetic studies were conducted on animal models to identify that how much drug reaches the plasma drug levels. And lastly, toxicity studies are recommended for the microemulsion based drug delivery systems. Apart from the test to be conducted on reconstituted microemulsion, there are other tests which are also required to be conducted on pre-concentrated microemulsion, that is solubility studies in of a drug in the PCM system, drug delivery performance studies equally as that conducted for reconstituted microemulsion, surface tension and viscosity determination as done for the reconstituted microemulsion. And apart from that, one of the important studies these days is instrumental texture analysis, which will give us firmness, index of viscosity of a PCM, and consistency of a PCM, that will directly or indirectly influence the formation of microemulsion. Now let us elaborate what are the different tests to be conducted on reconstituted microemulsion one by one. The face behavior, which is conducted using visual inspection, is conducted for the clarity of a microemulsion, and the clear, not turbid or milky white reconstituted microemulsion indicate acceptable formulation, whereas transmittance, that is quantitation of this turbidity levels, are to be conducted with the help of a percentage transmittance determination of a microemulsion, and is determined using UV visible spectrophotometer at a wavelength of approximately 560 or 630 nanometer. But these wavelengths are not specific. You need to determine the lambda max of a particular turbidity levels depending upon the microemulsion you are looking for. Now this image indicate that a ternary phase diagram is drawn between water phase soybean oil and 280 as a surface active agent, and we have obtained a small microemulsion area. You can see different images which will show a gel-like consistency, my emulsion type consistency, microemulsion area, but in this case we have obtained a small microemulsion area which is visibly clear but bluish in color. Phase contrast microscopic technique is one of the technique which will give illuminescence to the you can say samples which are transparent or translucent in nature. This technique was first developed in 1934 by Dutch physicist named Fritis Jernicke. It's a contrast enhancing optical technique. It is particularly initially it is developed for the visualization of you can say living cells, microorganisms, thin tissue slices, but now we are also extending this phase contrast microscopic technique to visualize the droplet size of a microemulsion. In principle, this phase contrast microscopic technique is an optical mechanism to translate minute changes in phase into respective changes in amplitude, which can be visualized as differences in image contrast. So for this particular purpose, what we will take is we need a specially designed illuminating material, as it is a source of light. So here we use it as a tungsten halogen lamp, which is directing through the collector lens and focused on a specialized annulus and positioned in the substage condenser front focal plane. And this particular light is then used as a wave front passing through the annulus illuminate the specimen, either passing through undeviated or diffracted. Now we have a diversion of light where we have undeviated or a diffracted light, and then these are retarded in phase by structures and phase gradient present in the specimen. So undeviated and diffracted light collected by the you can say objective piece, which is placed above the specimen sample, just above the specimen sample, is segregated at a rear focal plane by a face plate, and the focus at the intermediate image plane to form the, you can say, a final phase contrast at just at the surface is observed in the eyepiece, and that is why we will observe highly illuminating, you can say, a material in case of phase contrast microscopy. Now the luminescence which are produced by tungsten halogen lamp is sent to the condenser, and that condenser illuminates specimen, and this specimen is then project the light to the objective, which is then converted into two parts with the help of face plate and as diffracted light or a direct light, and then the image is available as a contrast image through this microscope. Freeze fracture TEM is another technique which describes the breaking of frozen specimen to reveal internal structures, and these freeze etching is then sublimation of the surface ice under vacuum to reveal details of the fractured face that were originally hidden. Therefore, this investigation is utilized for microemulsion, formulations, cell organelles, membranes, or emulsions. Freeze fracture TEM. For the freeze fracture TEM, the samples are prepared in a specially, you can say, design technique. Since the name is freeze fracture, so we have to freeze the sample, and then we'll send it for the TEM analysis. That is the principle behind it. In brief, what we can say is 1 mm cube sample size is generally taken. For the case of liquid samples like microemulsion sample, we'll sandwich the sample between the thin metal sheets, forming a sandwich between each other, and then these samples are frozen. And these frozen samples at around -100 degrees Celsius and -150 degrees Celsius, and then these frozen samples and undergo you can say a microtome treatment. After microtome treatment, then these samples are then evaporated or sublimes, and after sublimation we will obtain, you can say, cavities of the spaces in the on the slide, and then these slides are then visually sent to that of the TEM analysis for getting a replica of TEM images, and these deeper layer features can be visualized with the help of these TEM analysis. This image revealed different scheme for the freeze fracture TEM analysis. You will prepare first a sample, and then that sample is freezed. After freezing, it is fractured, then etching, evaporation of the aqueous or watery layer, and then cleaning so as to obtain good images of freeze fractured TEM. This is an example showing that how is the shape of the image, shape of the microemulsion, that is nanoemulsion, visible using freeze fracture TEM. The next part is physical characterization, and for physical characterization, first is particle size analysis. For the determination of particle size, there are different techniques. We can have Taylor dispersion analysis, dynamic light scattering, nanoparticle tracking analysis, resonance mass measurement, laser diffraction, spatial filter, velocimetry, automated imaging. Among these techniques, the only techniques available with us is zeta sizer, which will give us a particle size of ranging from 0.1 nanometer to that of the 10 micrometer size. And that is why this technique is utilized in determining the particle size of a microemulsion. Although there is another technique based on laser diffraction is available, but which gives a sensitivity in the range of 10 nanometer to that of 10 millimeter size. That is the reason that we are preferring dynamic light scattering technique. The dynamic light scattering technique is used to measure particle and molecular size. This technique measures the diffusion of particles moving under Brownian motion, and this movement is converted into size, that is particle size, or particle size distribution by the application of Stokes-Einstein relationship. This image shows a correlation between percentage particles to that of diameter of the particles. This image is obtained when we use zeta sizer for the determination of particle size. At the peak of this bell-shaped graph, we have a mean particle diameter, and then the corners of the bell-shaped graph is called as a polydispersity index. And this polydispersity index gives us an idea of the distribution of the particles in a given sample of microemulsion. Polydispersity index, if we analyze, is a measure of particle homogeneity, and it varies from value of 0 to that of 1, and we can say the value closer to 1 indicates narrow size distribution of the formulation, and we always wish that our microemulsion should give PDI near to 1. This image is the plot which we have obtained after giving the sample to a zeta sizer. In the first row of this, we obtained a temperature at which we have obtained a particle size analysis, that is at around 25 degrees Celsius. In the second row, we obtained a mean diameter, that is 133 nanometer. PDI is 0.188, and the number of peaks which are appear will give us a detail on peak 1, peak 2 or peak 3. Since in this sample we have obtained peak 1, which has a size of around 145 nanometer with 100% intensity, and width is around 28.16 nanometer. So this is the way how we can analyze the sample which were generated using zeta sizer. Now this is another image of another sample of microemulsion. However, in this sample there is a 3 peaks which are obtained, and the particle size is different for all the 3 peaks. You can see in the second column, peak 1, peak 2 and peak 3 varies from different size proportion, and that is why 3 peaks are originated. And at the same time, since we have 3 different peaks, and that is why we have a PDI near to 1, that is around 0.533, and this is the reason this microemulsion system is not considered as a good microemulsion. Now these are the 2 images of zeta sizer. In one image we have a sharp peak, and this peak constitute a good microemulsion because PDI is approximately 0.310, whereas in the second image you can see 2 peaks which are originated. Therefore, such type of systems are not considered as a good microemulsion. For the determination of thermodynamic stability of a microemulsion, the 3 main parameters, that is cloud point, or freeze-thaw cycle or heating cooling cycle, that is also called as a heating cooling cycle, or centrifugation method are quite useful. The method which is cloud point determination is just you can say prepare a reconstituted microemulsion in the ratio of 1:100 in distilled water, and then it is you can say cloud point was determined as a temperature at which there is a sudden appearance of cloudiness. Both visually it can be determined, as well as we can quantitate it with the help of transmittance determination using spectrophotometric method. In conclusion, from these findings is as and when the higher the you can say cloud point, that is temperature, higher the temperature, higher the stability of a microemulsion. Another method for the thermodynamic stability is centrifugation method. In centrifugation method, we prepare 1:100, you can say a dilution of reconstituted microemulsion, and then it is subjected to centrifugation at around 6000 RPM for 30 minutes and observe for any phase separation or phase clarity. You can visually it can be observed. In case of heating cooling test, it is also called as a freeze-thaw cycle. In this test, we take that 6 heating cycles as well as 6 refrigeration cycles are to be given to a reconstituted microemulsion. Heating cycle may be at 45 degrees Celsius and refrigerator cycles at 4 degrees Celsius. And for each cycle we'll give 24 hours time period, and if the microemulsion is stable, then it will not give phase separation by when it is centrifuged at 6000 RPM for 30 minutes. So this is how we can get a heating cooling test done. A microemulsion is said to be thermodynamic stable when the cloud point is as high as possible. Centrifugation is given by a no phase separation, and during freeze-thaw cycle analysis, a clear solution is obtained even at 4 degrees Celsius and at 40 degrees Celsius. Rheology is a parameter which will give us the viscous nature or flow property of a microemulsion in the form of a pre-concentrated or a reconstituted microemulsion. Therefore, rheology determination is one of the very important factor. Rheology is estimated using cone plate method or cup plate method. Pharmaceutical microemulsion showed non-Newtonian behavior with yield point, whereas increase in temperature measured values of yield, stress and viscosity decreases. Therefore, we could say that microemulsion prepared by non-alcoholic co-surfactants showed Newtonian behavior. These are the images which are taken from research papers. They indicate a correlation between viscosity and speed. An intense dependence of viscosity on the shear tension was clearly visible from these plots, and it was evident that the composition of a microemulsion will definitely affect the rheological property of a microemulsion. Surface tension is another one of the important characterization of a microemulsion, which can be observed for both pre-concentrated microemulsion as well as that of the reconstituted type of microemulsion. To know what is surface tension is a molecule of a liquid attracts the molecules of you can say surrounding that particular liquid, and then the molecules which are just below it, they are also attached with the upper layer of the molecules. So now these molecules are balanced with each other so as to create a equilibrium with each other. So when these molecules are on the surface of it, they are attracted by the molecules below and by the lateral ones, but not towards the outside. So the surface tension expresses the force with which the surface molecules attract each other. And this surface tension can be determined by two methods. One is stalagmometer method and another is tensiometer method. Surface tension determined by stalagmometric method is using drop volume method, and for this purpose several drops of a liquid leaked out of a glass capillary of the stalagmometer are weighed, and then we are going to determine a surface tension using a mathematical expressions. Another method for the determination of surface tension is tensiometric method, and this tensiometric method is based on the principle of that ring, and that ring is just placed over the surface of a glass, and then different weights were applied, and the force required to just pull the ring is noted down using this tensiometer. This is the actually figures which shows how duonoid ring tensiometer looks like. The steps which are involved are, we just place a ring just over the surface of a water or a liquid, and then the force is applied with the help of a circular disc attached to it, that is called arm adjustment behind it. With this arm we just slowly increase the force, and the force which is necessary required to pull the liquid with the help of this ring is noted down using this disc. The surface tension determination is therefore one of the very important behavior for pre-concentrating microemulsion. The correlation of surface tension with the surfactant concentration gives critical micellar concentration, and this CMC determination will help us in choosing surfactant concentration and to predict stability of microemulsion. The lowest surface tension of microemulsion is an indicator of high solubilizing capacity of a drug and stability and lower self-emulsification time. The morphological evaluation of a microemulsion is conducted with the help of a transmission electron microscopy. This TEM analysis is conducted on microemulsion to determine the shape of particles, droplets, and that can be conducted at higher resolution and to give a point-to-point correlation between the droplets of the microemulsion. The procedure for the TEM analysis is 0.5 ml of a drop is put on a copper electron microscope grids, and then 0.5% weight by volume aqueous solution of phosphotungstic acid for 30 minutes is exposed, and then the sample is dried, and dried samples were then subjected to transmission electron microscope for the catching of images using this instrument of oil droplets of a microemulsion. These are the different TEM images which are obtained when a solid self-emulsifying drug delivery system is reconstituted in a water, and TEM images were taken. You can see a circular spherical structure like materials as observed in image A, image B and image D, whereas a clogging of a solid carrier is observed in image C. This image is the circular shaped oil droplets of microemulsion, and the image clearly says that the microemulsions are formed, and these oil droplets of a microemulsion are spherical in shape. In the second image you can see a dark internal structures which shows that the drug is entrapped inside these oil droplets. Now the electrical properties of a reconstituted microemulsion are determined by zeta sizer. The zeta sizer will clearly give us three parameters. One is called zeta potential, another is conductivity, and third is mobility. Zeta potential of microemulsion is nothing but a magnitude of zeta potential gives an indication of the potential stability of the colloid system. If all the surface of oil droplet in suspension have a large negative or a positive, you can say a zeta potential, then they will tend to repel each other and there will be no tendency of the particles to come together. If the particles have lower level of zeta potential value, then the particles coming together and flocculate, and therefore there is some, you can say, a value which is available in the literature which will predict the stability of microemulsion, that both the, you can say, oil droplets are separate each other. So we suppose that zeta potential should be in the range of plus 30 millivolt to that of the minus 30 millivolt. In that range we will predict it as a stable, you can say, zeta potential of a microemulsion. Particles with zeta potential more positive, then you can say positive 30 millivolt, or negative then minus 30 volt are normally considered as you can say a stable microemulsion. This is the image which is obtained from a zeta sizer which is on zeta potential mode, and the graph is between zeta potential in millivolt versus total counts. The peak obtained is not sharp, and therefore the instrument has recognized these peaks as peak 1 and peak 2. The overall zeta potential of this particular sample is minus 22.1 and the conductivity is 0.0995 milliseconds per centimeter. Another instrumental method is instrumental texture analysis. This texture analysis is to be conducted on pre-concentrated microemulsion using two probes. One is back extrusion and another is cone penetration method. Using back extrusion and cone penetration method, we have obtained firmness, consistency, cohesiveness, index of viscosity as the descriptors of rheological behavior of these samples. The maximum positive force of extrusion is determined as firmness. Area of that curve is consistency. The maximum negative force due to back extrusion is treated as cohesiveness, and negative area of extrusion is index of viscosity. Utilizing cone penetration method, we obtained work required to accomplish penetration was calculated from the area under the curve and is known as work of shear. This image is image of texture analyzer fitted with back extrusion ridge attachment. This is the image of attachment whereby the probe moves to and fro, that is up and down, with the help of an instrument, and we obtained parameters such as firmness, consistency, cohesiveness, and index of viscosity. To define firmness, firmness is a measure of maximum force that is required to extrude a sample from a concentric annular space between the plunger and the container, whereas the work of shear is the mean area of force versus time plot that is generated during the firmness studies. These graphs are generated when a sample of pre-concentrated microemulsion is given utilizing spreadibility probe, and force versus time correlation is generated. The peak at the positive side of the graph is work of cohesion, and that at the lower, that is negative part, is work of adhesion. Subtracting work of adhesion with work of cohesion will give us work of shear. When back extrusion ridge probe is utilized in the determination of different parameters of pre-concentrated microemulsion, these are the force versus time plots which are obtained. The positive side of this graph is work of cohesion, and negative side of this graph, that is lower portion, is lower to x-axis, is work of adhesion, and subtracting these two will give us work of shear. And we can also compare different other parameters like firmness, consistency, cohesiveness, and index of viscosity from these graphs. So students, let's see what we have learned from this module. The stability of microemulsion is predicted by testing its electrical properties, morphological properties, as well as physical properties of the microemulsion. The phase behavior of microemulsion is a direct method to ensure particle size and shape of oil droplets in a microemulsion. One of the very important thing is Zeta potential, which will directly predict the stability of the microemulsion, and Zeta potential should be minus 30 millivolt to plus 30 millivolt. And lastly, the characterization of microemulsion is important for ensuring good and acceptable microemulsion formulation.